with Luciana, and I haven't got my piece of paper, but you will tell us. And as yesterday, we will kind of keep questions till coffee, so hold on to your questions. So, digital twin platform for CAR-T dosimetry. Thank you.
Yeah, there we go. Thank you for giving me the opportunity to be here today. My name is Luciana. I'm a cross-disciplinary fellow at the CRUK Scotland Institute up in Glasgow. And that means I'm a physicist by training, a theoretical physicist. I come from loop quantum gravity and black holes and particle creation. And now I've transitioned to biology. So 5 years ago, when I decided to transition, I didn't know what exactly biology was. I knew I wanted to work with cancer because all my grandparents died of cancer. Many of my friends died of cancer since very early age. Even my dog and my hamster died of cancer. So I knew I wanted to do something with cancer. I didn't know what it was, so I spent a lot of time trying to learn what cancer was. And I think the first thing I've learned about cancer is that it's a very clever disease, and it finds a lot of mechanisms to hide from our body, right? So our immune system can't find it, so there's no way to fight against it. And even when we apply some therapies, it finds a way to come back. So another thing I've learned is about immunotherapies, and particularly CAR T cells. That was a before and after for me. So CAR T cells, for those who are not familiar, are just our T cells that we take out of the patient. We put some receptor, and that will allow our CAR Ts, our T cells, to find the cancer cells and kill them. Not only in the primary tumor, but if there is any sort of metastasis in our body, they will find it and they will kill it, ideally. That's not the case always. So it doesn't work in every patient. And even within the same patient, they will work in some places and maybe they won't work in some other places. And they tend to get exhausted quite easily. So we need a lot of research there because I feel like they are like the therapy of the future, but we still need to learn what's going on there. So in a very naive way, when I was transitioning, I was like, oh, I would like to create a tool in which we can predict and understand the tumor of each patient and then apply the therapy in the computer before applying it to the patient. I didn't know at the time that was called digital twins, and that's why we're here today. And that will help the clinicians in the decision-making process. So at that time, my supervisors and I had a discussion about what to do, and we decided an agent-based model would be like a great starting point. So we developed an agent-based model that allowed us to measure the interaction between cancer cells and CAR T cells. And then we can track how the different cell types evolve during therapy and how many doses do we need and how often. And I don't have time, I have like 2 minutes now, so I don't have time to go through the results of the agent-based model, but it was published last year. I'm very proud of that publication considering it comes from a very, very, very far away field. But if you have questions, please come to my poster. And I think that what I've learned is we need to calibrate models, right? So we need data. And the first thing is we don't have enough data. Yes, we are surrounded by data, but quantity doesn't mean quality, right? And sometimes the data we need to feed our parameters are not the ones that are out there. And second, there are some biological mechanisms that are still unknown. And coming from a very exact science as physics, it was kind of challenging for me to understand how can you work without knowing what's going on there? But that's good because it's a really good niche to grow, right? So after that, I got my fellowship last year. So I've been working, putting together this kind of hybrid lab in which I combine dry lab, which is the agent-based model. Now that I have my fellowship and resources and people with goodwill to share their work and resources, we're going to run experiments to Two things. First of all, understand some biological mechanisms that we are curious about CAR T cells, like why do they get exhausted? Why they don't penetrate the tumor? And that we're going to use a back and forth to feed the model and then use the model to inform. But as I said, I want this model to reach the clinic. So we've been working together in a collaboration with Google and Princeton and the CERN to use my model as an engine in this new platform they have called Biodynamo. So it's called Cartopia now. And the idea is to make it more accessible for people without training in coding and faster. So of course, this is not a one-person work. It's a massive group of people that gathered together the last year, a lot of institutions collaborating, and I'm just there in between trying to bring everyone together and talking to everyone. It's not easy to talk physics, math, biology. Communication is key here. So why am I here today and why now? First of all, this is kind of the platform for my lab. I'm an early career, I'm a fellow, so hopefully I'll be a PI soon. So feedback is always welcome. I know many of you have been there already, so welcome. Feedback is welcome. And second, I'm going to generate a lot of data set. And coming from a country in which we have no resources at all, I'm here because people share what they know and what they have. So all I'm generating here, code and data, it's going to be open, right? So if you feel that you can use something of these, or if you think that maybe adding one extra marker in my panel will help your research, please let me know. I feel we should support each other, particularly early careers. So send me an email or approach here or somewhere and just let me know. So thank you very much, and I'm very much looking forward to talk to you.
Thank you.